Hello, everyone, and welcome to my talk. I'm Christine, and I'm a data scientist here at PNNL, but based out of the Seattle campus. So it's great to be here at Richland, meeting some of my colleagues for the first time since I started work in the pandemic. And I'm here to talk to all of you lovely folk about a tool that I've been developing here at the lab called Snekmer. Now, many of you here are probably already familiar with the existing methods for protein function annotation. And these methods can include similarity search via tools such as BLAST, which stands for the Basic Local Alignment Search Tool. It's my mic. Okay. Alternately, for functional annotation, one can perform multiple sequence alignment followed by comparison against something like hidden Markov models using tools such as Hammer. However, these traditional methods tend to involve pairwise or multiple sequence alignment, and as such can be rather time and resource intensive. And additionally, sourcing appropriate HMMs for modeling your systems can be difficult to both source and build. Our solution to these problems is the representation of protein sequences using K-mers. And so K-mers are a way to represent a protein sequence by counting the substituent sequences of length K. And what this allows us to do is capture sequence similarity between partial or distantly related sequences. And K-mers additionally confer the advantages of being relatively lightweight and thus having lower computational overhead versus something such as a multiple sequence alignment. Additionally, K-mer representations conveniently come in a vector form, wherein the vectors represent the number of times certain K-mers have been counted in the parent sequence. And vector forms are amenable to transferal to machine learning methods. So this introduces an opportunity for us to represent a sequence quantitatively in a vectorized format that captures structural information. Now, K-mers have already been widely used in genomics to represent nucleotide sequences, but as you can imagine, a key disadvantage of using K-mers for protein sequence representation is that there are unfortunately 20 amino acids. And so you can imagine that as we increase the length of K-mers that we in our analysis, that scales with 20 amino acids and quickly can become intractable, particularly if we're considering longer K-mer lengths. And sometimes those may be needed to capture important structural motifs. So to address this issue, we have developed an approach that we've termed amino acid recoding, which is relatively self-explanatory, but I'll explain in a little more detail. What amino acid recoding, or AAR, does is groups amino acids by, for instance, chemical properties or other measures of similarity. We've also grouped some amino acids according to certain substitution matrices, such as the MIQS matrix substitution. Incidentally, that stands for matrix to improve quality and similarity search. Fun factoid. And what this does is it allows us to reduce the sheer size of our K-mer space while still preserving some chemical information that may be represented by that original amino acid. So I'm all about building tools and software, and in building a tool that invokes this type of functional annotation, we envisioned a modular workflow that combines the AAR approach with the K-mer approach. That's also suitable for high throughput analysis. Our workflow at a high level, we envision beginning with applying our described amino acid recoding to a particular protein sequence according to whichever scheme you desire. And now taking that recoded sequence and applying K-merization, or the generation of these K-mer frequency vectors. And what that does is just goes through the sequence, counts the K-mers that compose that sequence and collates those into a frequency vector. We can then feed that vector into machine learning models to quantify similarity or else model sequence similarity between proteins based on their K-mer profiles. To do all of this, we've implemented all of these high-level ideas into a workflow slash tool known as Snekmer. And Snekmer is fully open source, available on GitHub publicly at the PNNL CompBio GitHub repository, and is written in Python and thus benefits from this rich ecosystem of bioinformatics and data science packages. As the name might also just suggest, Snekmer is built upon Snakemake, which is a Python-based workflow management system, and that confers many advantages to our system, such as innate reproducibility of the system, scalability across diverse systems from single core, multi-core, cluster, and cloud computing environments, as well as modularity in how the steps of the workflow are implemented in the first place. So in Snekmer, we execute our envisioned workflow via these self-contained modular steps. And for those of you familiar with Snakemake, these are essentially Snakemake rules. Well, they are Snakemake rules. What happens here is that first we start with our input files. Those are detected and processed via Snekmer and then fed into what we call a K-merization step. And here is where the magic happens. We perform our recoding according to whichever user specified parameters, apply K-merization via the user specified K value, and build those frequency vectors. And then we have here sort of a choose your own adventure type of selection for the user, depending on the application. And so if the user has, say, a set of pre-annotated protein sequences and would like to use those annotated sequences in order to develop annotations for unknown sequences, they can take those curated protein families and feed that into the top workflow, which is the Snekmer model workflow. And here what we do is we then take those K-mer frequency vectors, use those to develop frequency probability scores based on the prevalence of certain K-mers within given protein families. And from those, we build machine learning models, which essentially are classification models for whether or not a given protein sequence belongs to a given protein family. The user may also not have these nicely curated pre-annotated protein families and may be more interested in the relationships between different protein sequences based on their K-mer profiles. And for that, we have the second workflow, which is Snekmer cluster. And in that case, we use Snekmer to apply a number of available unsupervised clustering algorithms and generate clusters that group these proteins by similarity based on their K-mer profiles. We also have a third operational mode of Snekmer known as Snekmer search. And what that does is it takes those pre-trained Snekmer models and can then apply those to annotate unknown sequences. And the output for the user is our family probability scores based on the adherence of those unknown sequences to our expectations in terms of expected K-mer profile. And so it goes through all this scoring model pipeline and then essentially searches that model space for likely annotations. To validate our pipeline, we evaluated a curated set of annotated protein sequences from proteins belonging to the nitrogen cycle. And this consists of 33 protein families and about 5,500 sequences in total. And those were previously annotated using hidden Markov models. And as a further step to the data curation process, we additionally separated certain subfamilies with divergent, certain families with divergent subfamilies, sorry. And then as you can see an example of the nitrogen cycle here and the involved families. It's a little small, I apologize, but essentially each of these families is involved in certain steps in the nitrogen cycle. And this is actually illustrated by Jason McDermott here at PNNL. So we ran Snekmer model mode across 6 recoding alphabet schemes, as well as once with no recoding scheme, just all the amino acids, and varied K in order to evaluate the performance of the resulting classifiers. So the models were evaluated via 5-fold cross-validation, And the performance of the models is summarized here using the AUC ROC, which stands for the area under the curve of the receiver operating curve. And essentially what, for those who aren't familiar, an AUC ROC of 0.5 corresponds to random chance, whereas an AUC ROC of 1.0 corresponds to perfect classification. And so what you'll notice here is that one, we found that the AUC ROC is greater than 0.9 in more than half of the alphabet and K combinations, thus closely mirroring the original HMMs. And furthermore, we found sort of an interesting relationship between the alphabet and K configuration as well and the performance of the classification models. So you can see here on this figure, if you focus on the figure on the right, sort of along the diagonal, there's a Goldilocks zone at a moderate alphabet complexity and moderate K-mer length where the models seem to perform best. And this makes sense if you think about it. Consider starting at a very low complexity alphabet. So here at the simplest, we have hydrophilic and hydrophobic amino acids. When we recode into that 2-character alphabet, we have a very simple alphabet encoding. And when the K length is very short, those K-mers are highly non-specific and they're likely to occur in many different types of sequences, regardless of their function. And so you can see that the resulting classification models are correspondingly a lot less accurate. In fact, the shortest K-mer length for the hydrophilic hydrophobic alphabet actually performs no better than random chance on average. Whereas as you start to increase that alphabet complexity and K-mer length, you hit this optimal zone where now the K-mers are optimally complex in order to capture all the pertinent structural information that encodes potentially the protein function. And there the classification models start to perform the best. And then you can overshoot that optimal zone where now the alphabet complexity is very high and your K-mers are very long. And in this case, now you have very long, highly specific K-mers. And what that means is that you're fitting your models to K-mers that may not be representative of that family set. And so the performance of those resulting models actually begins to decrease again. We also evaluated Snekmer, we evaluated the nitrogen cycle families using the Snekmer cluster mode and found that the resulting clusters work well at recapitulating these families. And furthermore, we observed similar trends as I described in the Snekmer model mode in terms of how well the clusters capture the families, as well as a little lightly as well, the relationship between the selected alphabet and K value versus the performance of the resulting clusters. So while our results from the test case of nitrogen cycle families were very promising, we envision that the end user will not typically have neatly curated sets of highly specific proteins to build models with. And more realistically, users may want to use Snekmer to analyze something like whole genomes with partial annotations and a much larger data set size. So to address this paradigm, we evaluated Snekmer against whole genomes. And in this case, we randomly selected 10 genomes from UniProt SwissProt consisting of around 47,000 sequences, and then used Snekmer's clustering mode to generate clusters based on the AAR K-mer approach. And lastly, to validate or benchmark our approach, we compared the clusters that were assigned via Snekmer's clustering mode versus a known state-of-the-art clustering method that's that you may know that's used in the field, known as MMSeq2, which stands for many against many sequence searching. And we use this in order to sort of benchmark the compare the resulting clusters and see how similar or how dissimilar they were versus an already existing method. And I'm summarizing the results here. The numbers here on this plot indicate the number of protein sequences that were assigned to a cluster using either Snekmer or MMSeq2. We found that there was similar performance between Snekmer and MMSeq2, and also found that the either clustering method essentially assigns about 50 to 60% of these sequences into clusters. However, those 50 to 60% actually don't perfectly overlap. So there are a fairly sizable number of sequences actually that MMSeq2 does not assign into a cluster that are assigned into a cluster via Snekmer. And so we see an immediate application of Snekmer to the augmentation of assignments, augmenting existing methods with Snekmer. And for instance, in this particular case, we're now increasing the coverage from that 50 to 60% to something closer to 85% of sequences now having an assigned cluster. Excitingly, we find that Snekmer identifies clusters that aren't found via MMSeq2. And this is the subject of ongoing work where we're working on evaluating whether or not these clusters are false positives or actually contain interesting information. But I think the fact that our clustering method based on the AAR Kamer approach generates unique clusters that can potentially add new information or be used as an additional annotation tool is pretty exciting. And we're really interested in exploring that in more detail. So in summary, I've developed this tool known as Snekmer that takes the Kamer and AAR approach, and we've evaluated that using known protein families in order to evaluate its potential towards annotating new families. We have a fully working prototype available, and our publication is actually in revisions to be resubmitted very soon, which we're very excited about. And all the code is available online at GitHub with documentation as well. We have many future plans for Snekmer, including first, we are working on code optimization to facilitate its usage towards larger sequence sets, as well as working on making Snekmer a lot more user-friendly. We want Snekmer to be available to everyone and be a useful tool for biologists. And what that entails is adding some user-friendly interfaces. So we're working on making a web app version available, as well as integrating Snekmer as an app into K-Base, which is a sort of a data science platform for biologists where you can integrate different tools together. And it would be really awesome to have Snekmer as one of those tools available for biologists. And so with that, I'd like to thank all the people without whom Snekmer would not be possible. And in particular, I would like to thank Jason McDermott, who came up with the idea that became Snekmer and really led this work, as well as Bill Nelson for driving the application-based side of Snekmer, for data curation and helping us make Snekmer a better tool for the end user. And I also like to mention our funding sources like the Soil, SPD, SFA, led by Rob Egbert, as well as K-Base. And I'd also like to thank all of you for your attention. And I'd also like to shout out, if you're interested in knowing more about Snekmer, we have a demo coming up right now, pretty much. It's going to be in Vista, the other room. And so if you'd like to come see what Snekmer's all about and how to use it, please stop by our demo, and I'm happy to take any questions. Thank you.
Thank you, Kristen. Questions for Christine?
Hi, thank you for the talk. It was very interesting. I just have a quick question because I'm more coming from the structure world. And I know that MMSeq has been used in AlphaFold, and it seems that if you're determining new clusters, there is another level of information that most likely you're finding. And as far as in this, I understood that your K-mer definition comes based on basic properties of amino acid. And have you ever looked at the maybe smaller structural motifs as your K-mer, like vectors for further more detailed analysis, because now you have a lot of structural data. If you can kind of get small building blocks, structure motif, and use them as a k-mer vectors, that might be like, I don't know, like interesting thing to take advantage of. Maybe I don't know how the development goes, is it possible or not, but just from what I'm seeing, there is something extra that you're able to mine in your data, so it's great. Thank you.
Yeah, thank you. Yeah, that's definitely something, a direction that we're really interested in, is expanding the idea of Snekmer and being able to encode not just amino acids one-to-one, but having more complex encodings. So that's definitely sort of another future direction. We have a lot of ideas for the tool and limited manpower and time, but definitely that's a fantastic idea.
One question here.
So does it make sense to automatically select K? Do you do it also?
Actually, we've explored the idea of having some type of automated or semi-automated automated parameter selection, but right now we've created Snekmer as a sort of a flexible tool where the user can decide their own parameters. Different parameters are actually, I haven't shown those here, but can be better or worse at capturing different protein families in terms of the classifier models. And so we think that having that flexibility and leaving that to the user for now is probably more makes it so that eventually we can perhaps extend that into streamlined automatic parameter determination. But for now, no. We leave that open to the user, and they can choose to select, a very long K or a very short K, depending on whichever application.
We have time for one quick question.
Yeah. Thank you.
Can the user define a different alphabet?
That is something that we've actually thought we would love to include that functionality actually. And right now we only have those 6 encodings that I shared on the previous slide. And the user can also specify no encoding, but that's a subject of future work, definitely. We'd love to enable the user to specify their own alphabets.